So we're going to look at the cell cycle just in broad terms today, and then we will break it down into the more specific information later on this week. So to start out with, let's review why cells have to divide. If you are a unicellular organism, then every time you divide, it's also the same thing as reproduction, right? One cell breaking apart into two, and each of those cells are its own organism now, so that is essentially just reproduction. But in multicellular organisms, so plants, animals, and fungi, they're going to do cell division to grow, which is making more cells, making the organism larger. They're going to also do this to develop, so the process where where one cell divides into two, and those daughter cells, those cells that are now produced at the end of cell division, have a little bit different gene expression than what you started with. That is, they are more specialized and more like a particular tissue. So for example, your skin cells are different than your liver cells, and they're different because they have developed, right? They have differentiated or developed into such a way that they have different genes that work. Okay, so multicellular organisms, they are dividing to grow, to develop, and then finally to reproduce. And it's not going to be all cells that are reproducing. Instead, it's just going to be the reproductive cells in sexually reproducing organisms. In the case of asexually reproducing organisms, usually these are going to be not the case, because the entire organism as a whole is going to kind of chunk off and make its own organism. So multicellular organisms using cells to grow, develop, and reproduce. An important idea here is that prokaryotes and eukaryotes have very, very different genomes, right? The genomes are the complete set of genetic information that's in an organism. So every cell in your body has all of the genetic information to make every other cell type. Your entire genome is in every cell. And if you're prokaryotic, because they're so simple, they're small, there's not a lot of genes there, they're usually one piece of DNA that loops back around to itself. They're so simple, which makes cell division a lot easier. Eukaryotic genomes, conversely, are really big, and they're so big that they have to be packed together into chromosomes so that they don't all get tangled up. And because there's a bunch of them, it's going to change the process of being able to divide those cells. At this point, I've covered 1 and 2 on the lecture notes as an FYI. So we can take a look here at the process of cell division in prokaryotic cells. We start out with a simple prokaryote, not a lot going on. It doesn't need a huge set of genetic instructions because it's pretty simple. So the DNA that it has chilling there right in the middle in the cytoplasm and not in the nucleus, that DNA is going to start by dividing. That's going to give enough genetic information so that when the cell divides into two, each daughter cell, each cell that's produced at the end, has the genetic information that it needs to be able to survive. So when we're looking at prokaryotic cell division, we're going through the process of what is called binary fission. And so the first step, the DNA replicates, and then that cell overall is going to divide. And again, why is it so easy for prokaryotic cells? I mean, this is fast. They can do it in a matter of 10 minutes on the fast end to about half an hour, 45 minutes. So prokaryotic cells can replicate super duper fast. I mean, we're talking, we could be covered on Earth in miles deep layers of bacteria if they could divide as much as they wanted to and as fast as they could. So it's a really, really fast process. And the reason it's so fast is because that simple singular genome, or chromosome really, can be replicated, separated, end of story. I like to give the analogy of using a blueprint to make a building, and in the case of prokaryotes, we're thinking not a very complex building, instead just a little shed. So it's really one page of instructions in order to make this little shed. If I photocopy this and make now 2 sets of instructions, it's easy to split that off for 2 different individuals to be able to then build the shed. Same thing, DNA replicates and it's easily divided among 2 different cells, so cell division is pretty rapid. If we were to try this in eukaryotes, however, things get a little bit dicey because of the fact that they have chunks of chromosomes, right? So if we were to replicate this genetic information and then separate out the nucleus into two, and then eventually separate the rest of the cell, notice that you don't get a complete set of instructions in both nuclei. So what you'd end up with is one cell that has some information but not the other, and the other cell that has the rest of the information. So instead of complete sets, you're kind of left with a mess, and that's sad. Okay, so for eukaryotic cell division, we're going to go through a process either called mitosis or meiosis, and we'll look at the difference between those here in a little bit. In both cases, we're going to start off with the DNA replicating. I know there's not enough dots on your guided notes, just add an extra one in there. So the DNA is going to replicate, then we're going to have the nucleus dividing first, and then the rest of the cell dividing. And what makes it so complicated is because we've got these huge chromosomes that are chunked up. So instead of one piece of information, it's in 46 or whatever number of chromosomes the organism has. So those have to be sorted out, and that's going to be what ensures that each new offspring cell receives an entire set of chromosomes. Okay, so we're going to move on to looking at the process of, or at the cell cycle. And to understand this, I think it makes some sense to look at the human life cycle first. We can look at it linear, as a baby growing into a toddler, growing into an adult, making some babies, and then dead, right? Sorry, Grandpa. So that process, that's the human life cycle. And we can also look at this, instead of looking at it in a linear fashion, look at it as a circle, right? We start out as a zygote right after fertilization occurs. So you are one cell, and then you grow into an embryo, and then into a fetus, and then into baby and child and adolescent and grown up. And then as a grown up, these people here are going to produce these cells that end up being the next generation. And then when these fuse, this cycle repeats itself. So we can kind of look at it as a cycle like this. Brand new baby grows from an infant to a childhood through adolescence. And that is all childhood. That whole process is all about growing and developing and learning and, becoming ready for adulthood. So that's the human life cycle. And then it's really only this little piece here, puberty, adulthood, and pregnancy, that are going to be involved in reproducing. Okay, so now we can look at the cell cycle in the same way. A brand new baby cell here is going to start in interphase. Interphase is the phase where we're not actively dividing. The cell is not actively engaged in cell division. So we start off with a period of growth called G1. And that's exactly what it sounds like. The cell is just growing. During S, this is the phase that stands for synthesis, which really just means DNA replication. And then once the DNA has replicated, we know that that cell is going to need to divide, because now it's got twice as much genetic information. So G2 is all about preparing to undergo the process of cell division. And it's really only this M phase right here, this short little time during a cell's life, where it is actively dividing. So that's going to look something like this. I start with the cell. It's got a certain amount of genetic information in the nucleus. And so during G1, the cell is going to grow. But notice that the nucleus does not. So the cell isn't going to be able to continue to carry out all the functions that it needs to do, because the rest of the cytoplasm is so large. So it's going to go ahead and do some DNA replication to make 2 full sets of chromosomes. So the entire genome is replicated and occurs twice. During G2, it's final preparation to get ready for the M phase. But up to this point, the cell has not divided. So that's interphase, G1, S, and G2. During M phase, 2 processes happen: mitosis or meiosis, and cytokinesis. So during mitosis or meiosis, the nucleus divides, right? So we started out with one nucleus that had double the set of information, and through a very meticulous sorting process that we're going to look at later, that one nucleus divides into 2 that each looks like the original nucleus that we started with, or has the same type of genetic information. And now with 2 nuclei, that's going to allow the rest of the cell to be able to divide, and what we're left with is a brand new baby cell ready to go through interphase and move on in its life. So I think instead of in terms of chromosomes, I think it's really helpful to think of office supplies. It's just a lot more visually easy to understand. So when we looked at our dragon chromosomes, we knew that, like, for chromosome 1, for example, chromosome 1 had all the same genes but potentially different alleles. And so we called chromosome 1 that were different, we called them homologous chromosomes. And the same thing is going to be true here. This is showing you that there's 2 expo markers, one that presumably would have been inherited from 2 different parents, and they have the same general information, they have the same function, but they're different versions of the same thing. So homologous chromosomes have all the same genes, but potentially different alleles. Okay. And so in this case, I have 4 completely distinct office supplies. So we're going to say N equals 4. That's kind of the nomenclature that we're using, where N is different chromosomes. So we have the expo markers, we have the scissors, we have the lanyard, and we have the ruler. And for each one, we have 2 copies. So this is called diploid. So in human bodies, we are N equals 23, and we're diploid. The normal cells in the body are diploid, so they have 2 copies of each of the 23 different chromosomes, making a total of 46. I wrote all that, but it's back there. Okay, so normal human chromosomes, during S phase, they're going to replicate the DNA, and now you're left with twice as much. So let's go back to that. Here we've got our cell. Or what is being represented by a cell, which is N equals 4, 2N, a diploid cell. So 2 copies of each of the 4 different office supplies. During S phase, or the analogy for it, each of these would replicate. And during the process of mitosis, they would separate into 2 separate nuclei. So 1 nucleus here and 1 nucleus here. And so what you should notice is that at the beginning, I started with 1 diploid, N equals 4 cell. And at the 2, now I have 2 diploid, N equals 4 cells, because my new cells have 2 lanyards, 2 scissors, 2 rulers, 2 expo markers, 4 different chromosomes, 4 different office supplies, 2 sets of each. So mitosis looks like this. You start with, and you can go ahead on your packet and cross out numbers 6 and 7, because it is repeated in the table, and I think the table makes more sense. So you are filling out the table, which is number 8, and what you're starting with is a diploid, or 2N cell. During the process of mitosis, each chromosome gets separated 1 time. You end with 2 diploid cells. They're identical to each other and identical to what you started with, and this is what are making the body cells, or your somatic cells. So I remember this as the process that happens from my head to mitosis. Mitosis, get it? Anybody? Okay, moving on. So now let's take a look at meiosis, okay? If we were to take these cells, this cell right here, for example, this, let's pretend this 1 nucleus was in a cell, and it was a sperm or an egg, and it got fertilized. When you took that, fused it with another cell, now you'd have twice as much genetic material as what you need. So meiosis is really all about cutting that number in half so that when 2 cells fuse together, you can go back to being diploid. You'll see what I mean here in a sec. So we're starting out the same way. This cell is 2N, N equals 4. We've got 2 different versions of the expo markers, the rulers, the lanyards, and the scissors. We're going to go through S phase, so we're replicating all of this just like we did before, right? During interphase, we go through the period of growth, where the cytoplasm grows, synthesis, where the DNA is doubled, that's this here, G2, and now during the process of meiosis, we're going to divide everything up, not once, but twice. That's going to lead to 1, 2, 3, 4 separate nuclei. So take a second and compare these together. They're not identical, but they're pretty close. So each one has an expo marker. Each one has scissors. Each one has a ruler. And each one has a lanyard. And in chromosomal terms, this would be a cell that had one copy now of chromosome 1, one of chromosome 2, one of chromosome 3, 4, etc. So what we're going to call this, we're still n equals 4. We still have 4 different chromosomes here. But now none of them are with their homologous pair. Instead, we are at 1n or haploid. So the purpose of meiosis is to start with a diploid cell, but then by separating out the genetic information twice, separating the chromosomes twice, you're going to end up, instead of 2 cells that are identical, you have 4 cells. They're no longer identical to each other. They're each unique, and they're haploid, meaning they have one copy of each chromosome. And the way that you remember your gametes is there's 2 kinds of gametes that are involved in meiosis, and they are the sperm and egg cells. So meiosis is the production of sperm and egg cells, or gametes, right? So they have half the genetic information, so that when fertilization occurs and a sperm meets an egg, you go from being 1n for the sperm, 1n for the egg, and guess what? 1 plus 1 is 2. You're back to being diploid, and that becomes the baby. Yay, right? And we can go all the way back here and see that we already saw that in the human life cycle that I didn't at the time expect you to understand completely. I guess I could find a different button to go back. Here we go. The adult male is making sperm. The adult male is 2n. The sperm is n. The adult female is 2n, and she is making eggs that are n. N plus n is 2n, and we've made a zygote, or a tiny itty-bitty baby that is going to grow up and continue to be diploid. So that is the end. If you've got questions, hit me up. Otherwise, don't forget to hit the subscribe and like button and the bell so you can get notifications when I post a new video. Woo!